In the heart of every living cell, an intricate dance of life unfolds, a dance that involves the conversion of DNA, the blueprint of life, into RNA, a messenger carrying the instructions for building proteins. This process, known as transcription, is a fundamental part of biology. Transcription, in essence, is the first step of gene expression, where a particular segment of DNA is copied into RNA by the enzyme RNA polymerase. This process is vital in creating the proteins that make up the structure and function of our cells. Think of it this way: if the DNA is the master architect's original blueprint, then RNA is the worker's copy, used to build the structures that keep the cells functioning. Now let's delve deeper into the step-by-step process of transcription. Firstly, initiation takes place. This is when the RNA polymerase binds to a specific region of the DNA called the promoter region. This region signals the start of the gene to be transcribed. It's like the starting line in a race, telling the RNA polymerase where to begin. Once the RNA polymerase has bound to the promoter, it unwinds a small portion of the DNA helix, exposing the DNA bases in that region. Next comes the elongation phase. Here, the RNA polymerase reads the DNA strand and begins to synthesize the RNA strand. It does this by adding nucleotides, the building blocks of RNA, in a sequence complementary to the DNA strand. In this step, the RNA polymerase moves along the DNA strand, unwinding the DNA and elongating the RNA strand as it goes. Finally, we have the termination phase. This is when the RNA polymerase reaches a sequence of DNA called the terminator. When the RNA polymerase hits this sequence, it releases both the DNA and the newly formed RNA strand. The DNA rewinds back into its original double helix structure, and the RNA strand floats off, ready to be processed and used to build proteins. So to summarize, transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. This process starts with initiation, where the RNA polymerase binds to the promoter region of the DNA. The polymerase then unwinds the DNA and begins the elongation phase, reading the DNA and synthesizing a complementary RNA strand. Finally, the RNA polymerase reaches the terminator sequence, where it releases the DNA and the RNA, marking the end of transcription. The process of transcription is a testament to the incredible complexity and elegance of life at the molecular level. It is but a single step in the journey from gene to protein, but without it, the intricate machinery of our cells would grind to a halt. Remember, every protein in your body, from the enzymes that break down your food, to the antibodies that fight off infections, to the pigments that give your eyes their color, all started their journey to existence with transcription. In essence, transcription is the bridge from the genetic blueprint to the living, breathing reality of life.